This report brought to you by the Richmond Club, where investors and high-growth companies meet.
Good afternoon, ladies and gentlemen. Our first speaker is the President and CEO of MEI Pharma, Inc. Please welcome Dr. Dan Gold.
Thanks, Scott, and thank you all for coming. It's a pleasure to be here in sunny Toronto. We're an oncology-focused company based in San Diego, where the weather is a little bit different most of the time. So it's always a pleasure to come and see how the other half lives. I know the timing is short, so I'll get right to it. We are an oncology-focused company, publicly traded on NASDAQ. We strive to build a comprehensive oncology franchise. Currently we have 4 clinical oncology programs. All of them will be in the clinic within the next several months. By this time, in about 4 or 5 months, we'll have 6 clinical trials running. Near term, we have multiple milestones coming up in the first half, and we're trying to build an efficient business strategy. It's tough as a smaller company in biotech to come up with a business model that really works. You have the behemoth pharmas, and we all try and struggle to find an efficient niche where we can play. We're trying to build a model where we access interesting drugs that we think are important. We try and build value into those drugs. We own them all. And then we make the decision if it's worth doing a strategic partnership, either globally or regionally. And eventually we obviously hope to be able to take all of these drugs or future drugs across the finish line and sell to sales and marketing ourselves. We have a robust IP profile right now for all of our drugs. We have a coverage out to 2037 on several of them. And at year-end 2017, the end of December, we reported $42 million in cash in the bank. I have a good friend who used to be one of those star analysts on Wall Street in biotech, and we always used to lament that there's probably as many drugs that were failed by the companies that were developing them as there are companies that were failed by their drugs. And I think that's a really important point, because drugs have to be good. There's no question about it. A bad drug is going to fail. But how you execute the development of that drug, the strategy that you use, the trials that you design are equally important. And if you don't have those components, you will fail. In fact, in drug development, we still have about 50% of drugs that go into phase 3, the final testing ground, where a drug, you should have worked out all the kinks, and now the drug is just going through that final testing for approval, and 50% of the drugs still fail. So it's a daunting task, and you really need all points taken care of. So I won't go through this slide. I'll just point out our clinical strategy is run by Dr. Mass, Dr. Robert Mass. He was 12 years at Genentech before they were taken out by Roche. Bob has many, many years of clinical oncology experience. He brought the drug Avastin, which is one of the leading selling drugs ever in oncology, to the market. It's important how you deal with the regulators. Karen, Dr. Potts, is our Senior VP of Regulatory Affairs. Karen came to us from another company in San Diego, Trias, where they got approved an antibiotic, and antibiotics are not easy to get approved. The regulatory path for them is very, very tricky, and Karen was able to navigate that. On our board is Christine White, our chairman. Christine was the former head of Global Medical Affairs at Biogen IDEC. She's an oncologist by training. Tom Reynolds is the former Chief Medical Officer at Seattle Genetics, and Nick Glover, a fellow Canadian actually, who's currently CEO of Sierra Oncology. So we have a lot of oncology experience on the board, and not to mention Kevin Clemens, who was formerly the head of Global Oncology Sales and Marketing at Roche. So what we strive to do is to bring the best people we can into the company to carry out our mission, which is to develop drugs. Now, when I meet people and I tell them what we do, they said, "Oh, are we near a cure for cancer?" And I think that that's an obvious question. It's what everybody wants to know, and the answer really is there is no single cure for cancer, because there's no two cancers are the same. Years ago, we appreciated the cancer of the breast, the cancer of the lung, the liver. They're all different. And now, as we see with more and more precision medicine, even one lung cancer to another is different. Even within a patient, sometimes a metastasis is different from the primary tumor. So there is not going to be a silver bullet. But as we learn more and more, we can come up with therapies that oftentimes when they're combined, they have a very potent effect in the patient themselves, and hopefully in multiple different patients. So we've taken the approach of diversifying our portfolio, and I'll go through very quickly what these compounds are meant to do. And ideally, as we grow the company, what we would like to do is to find other compounds that will synergize with these compounds as we develop them, so we can take multiple hits on the same cancer. Because really, if you look at the common chemotherapy that's used for treatments, it's not one chemotherapy, it's multiple chemotherapy. So you have to come at a tumor from multiple different directions. So our lead drug, which is now in a registration study, is an HDAC inhibitor. We're all built with a blueprint, our DNA, but how that blueprint is translated into making a cell a pancreas or making a cell into a liver cell is all after the blueprint is laid down. And what we call that is epigenetic modifications of the genome. And prasinostat, our lead drug, is a drug that it interacts with certain processes where genes are turned on and off. Cancers are very good at turning off genes that they don't want on, because those genes would stop them from growing. So this is a drug that presumably, we believe, will help turn on some of those genes that the cancers have turned off and cause the tumors to die. So this drug is in a registration currently, and we're also studying it in a second indication. Our next lead drug will go into registration by the end of this year. It is a drug that disrupts the tumors. Tumor cells like their environment. They kind of get cozy. And so when you drive them out of their environment, they're not so happy, and oftentimes that can be very disruptive for their life. So this is a drug that works on cells of the immune system, the lymphomas, cells that live in your lymph nodes and in the blood, leukemias, and it disrupts their ability to receive signals from that environment, which are very nurturing, and causes them to kind of freak out and die. Versatility is the newest drug in our portfolio. We'll be taking this into the clinic very shortly. This is an interesting drug, and I'll get into the mechanism of action. And this is a drug, 344. This is our legacy drug. It's a very interesting drug. It targets the tumor's ability to generate energy. Tumor cells grow like mad oftentimes, or they grow very rapidly, and they consume energy quite a bit. They need a lot of energy to grow, and our drug 344 tries to disrupt that normal energy that the tumors need. And what you'll see is that all of our compounds, all of our strategy is to use conventional therapies in combination with our drugs, because most cancers are not cured yet, and so there are treatments that make the patient live longer or have less symptoms, but they're not cured. So we're always striving to add on to what we know to make the therapies more effective. So in the very near term, we'll be reading out data from the trial in this disease called myelodysplasia. We'll be reporting on that in the next couple of months, and if the data look good, we'll be moving on into a larger randomized phase 2 trial, and possibly phase 2/3. MEI 401 will be reporting data on a large trial that we're running currently, which will set the stage for initiating a registration study later on in the end of this year. We'll be initiating new studies with Versatility, again in B-cell malignancies, and we just announced last week, I think, that a first look at interim data from a study that's currently undergoing study in Spain in HER2-negative breast cancer with Avastin looked positive, and we're going to go ahead and continue that study, and I'll give you a quick insight into that as well. So prasinostat, we think this is the best orally available HDAC inhibitor around. It has a proven mechanism of action. It has better drug uptake and exposure in patients. We've evaluated well over 300 patients. We know what the tolerability and what the toxicities are. And most importantly, we've shown our data in AML to the FDA. And based on that data, they gave us what's called breakthrough designation. What that means is it's not approved. It just says they've looked at the data. They know the field. They say that the data are compelling. They're willing to work with you on a more regular basis to help you advance this drug to approvals as quickly as possible once you have your definitive studies. So AML is an acute myeloid leukemia. You have cells in your bone marrow that make red cells. They make the cells platelets that cause you to clot. They make cells called neutrophils, which fight bacteria. This is a cancer of the myeloid system. The average patient is about 68 years old. In 2018 in the United States, we expect about 20,000 new cases with about 11,000 deaths. The problem with this disease, it grows very fast. That's why it's acute. If it wasn't acute, it would be chronic myeloid leukemia, which is another disease, CML. The problem with this drug is that as the patient's population ages, the therapies that are available to them, which tend to be higher intensity chemotherapy, because the goal is to kill all the cancer cells and get you into a bone marrow transplant, a lot of people aren't suited for that. They have other comorbidities that just make them not suited for high dose chemotherapy. And so we need a typical lower intensive chemotherapy for a large fraction of these patients. Before these low-intensity chemotherapy regimens were developed, the diagnosis to death was about 6 months in these patients. Now it's 9 months. The big advance, you've 50% improvement, but yet patients are living only about 9 to 10 months on average. Some people live for years, but on average it's 9 to 10 months. So there's a lot that needs to be done for these folks, because as we all get more experience, I won't say age, as I get older, these diseases tend to creep up, and we need to be able to do something. Thankfully, not only are we getting more experience, we're more fit as we get older, and there are more people that are able to take these therapies. Before, you would never think of transplanting a patient after 65 years of age. Now they do 75-year-olds. So we're getting more smart, we're getting more fit, and we're able to take this, but there's still a large patient population that just can't. So we took to the agency data that we had done. We treated 50 patients with our drug in combination with the other low-intensity chemotherapy. Ours is not a chemotherapy, it's a targeted agent, it's an oral agent. We showed the agency that about nearly half of our patients went into complete remission, which means we can't find the disease anywhere. The survival of these patients at 1 year was over 60%, meaning that we've now, whereas we expect these patients to go for 9 to 10 months, they were on average going out 19 months. So we've more than doubled the survival of these patients, and we've put about half of them into what's called a complete remission. So the agency looked at this data and said it looks very good, and they gave us this breakthrough therapy. Based on the breakthrough with the agency and based on this data, we entered into a partnership of worldwide rights to Helsinn Pharmaceuticals. Helsinn is a Swiss-based pharmaceutical company. They are very, very good at sales and marketing, especially in hematologic oncology. Combined with our clinical development expertise, we make a very, very good team. We received $20 million up front, up to $440 million in residuals, plus royalties that can reach up into the 20% plus. And they also made a small equity investment in the company. From here going forward, they're responsible for 100% of funding it. So it took a huge weight off of our shoulders. We were looking at doing a registration study that was going to cost in excess of $100 million. And for a company of our size, that was a daunting task. So Helsinn, it turns out, became a very, very good partner for us. This is the trial mainly, essentially it's a global trial, 500 patients, half of them will get our drug, half will get a placebo, and the goal is to show improved survival. We expect that this trial should read out in about 2.5 years. There's a cousin disease called myelodysplasia. It's a sort of in between a cancer and a non-cancer, but it generally, if the patient lives, they will get AML. They morph into this disease. We're running a study right now. We're paying half of it. Helsing's paying half. We're seeing if this treatment is effective. We lowered the dose because these patients don't need as high a dose. They tend to be much higher functioning than AML patients, so that we don't want to give them the fatigue and the nausea that accompanies a lot of chemotherapies or a lot of cancer therapies. If we're right and that they are able to tolerate this drugs, then we'll go into a randomized study, and we'll announce the results of this in the next coming months. We hopefully our hypothesis is correct that these patients will tolerate well and they'll respond, and then we'll go into this randomized study, which would form the basis potentially of another registration study. At that point, Helston would be 100% responsible. So let me move on to the next drug, ME401. This is just to say, I'm sorry, there's other drugs that are being developed because this is a very hot target. In fact, Gilead has an approved drug called idelalisib, and the other competitive products that are currently under development all have similarities to Gilead's drug. Our drug is completely unique, and we think that some of these unique features make this a really first or best in class. Some of those attributes, our drug is very potent. You don't have to take much. And because of that, we have what's called a high therapeutic window. You don't have to take a lot of drugs, so the difference in how much you'd have to give where the patient would start to run into toxicity issues is pretty broad. And that also enables us to have rather high versatility for combining it with other approaches. In the Gilead case, when you develop a drug to take into human beings, or cancer patients in particular, the agency and your pharmacologists look at the data that you provide to them in animals, in this case dogs, and they say, okay, based on what you're showing me, you can start at dose X. So this is the dose X that Gilead was recommended starting, and this is the dose that they ended up with. So there were 250-fold above the dose of drug that caused effects in dogs. So they already were sort of pushing the envelope in terms of the side effects. And it turns out, upon approval, this drug has a lot of hair on it. There are black box warnings all over the label. They have a lot of difficulties with liver toxicities and infections. When we went to our pharmacologist, they said, this is the dose that you could start at. We chose to start here because we didn't think we needed to go higher because we have high potency in our drug. So this is what we call this therapeutic index, is the difference between the levels that we need to attain and the levels that Gilead needs to attain. We're treating a disease called follicular lymphoma primarily. It's a type of non-Hodgkin's lymphoma. It resides mostly in your lymph nodes. It will then go into the bone marrow. About 20% of all lymphomas in the United States are of the non-Hodgkin's follicular phenotype. It's slow growing, good news. Bad news, there's no cure. So patients live for quite a while with their disease, but eventually they will succumb. The average age is about 60 years old. We are also treating CLL, but that's not going to be a focus going forward. So we're currently conducting a phase 2 trial that will read out. We'll be presenting the data this summer. We've already established what the dose is. The 60 milligram dose, in fact, we've done what's called dose escalation. We've gone up threefold in the drug over steps, and at every dose level, patients respond equally well. So we're not leaving any efficacy on the table by using a lower dose. We didn't see any dose-limiting toxicities as we went up in dose. So we think we have a much more potent drug than the Gilead drug, and we think it'll be more tolerated by patients. And as this slide says, we plan on initiating a registration study in what's called third line. So these are patients who have received 2 systemic therapies previously and whose disease has now come back. This is a very rapid way of getting an approval for a drug. And we expect to start that trial sometime in the second half of this year, closer probably in the October-November timeframe. Finally, our drug Versatility. Not finally, we have one more to go. So there's a whole class of drugs that are called cyclin-dependent kinases, CDK. They're intimately involved in regulating a cell's ability to divide and make new DNA. They form complexes with what are called cyclins, and they regulate the ability of the DNA to replicate. I'm just going to point to this one protein. We call it MCL1. So as we develop normally, as human beings, there are built-in programs for cells to die, because like when in the fetus, the growing limbs, cells have to die so new cells can grow beyond them, so we can form digits, for example. So that's called programmed cell death. In cancer cells, they've taken advantage. So you can imagine that's a very regulated process. You don't want cells dying all the time. So you have molecules in the cells that say die, and you have a whole set of proteins that say don't die. And it's the interplay of those two sets of proteins that will dictate whether the cell lives or dies. In cancer cells, they've taken advantage of this process, and they've highly upregulated these proteins that say don't die. So there is a drug on the market, marketed by AbbVie. It's called venetoclax. This is a drug that was under development with Abbott and Genentech for probably 15 years until they were able to crack it. And what this drug does is, this is an important protein in telling a cell not to die. It's called BCL2. Venetoclax comes in, it knocks off this little yellow BH3 protein that allows the BCL2 to form a death complex and die. What happens, though, is there are other proteins that are sort of overlapping, and MCL1 is one of the most important ones. So where this little protein's trying to plug in and form these death complexes, MCL1 competes out. And in fact, what's been shown clinically is that patients who don't respond or who relapse after venetoclax have upregulated the levels of MCL1 to compensate for this inability of BCL2 to work. So our drug, venetoclax, actually inhibits the production of MCL1. So we have a lot of data, preclinical data, that shows that if you add venetoclax plus our drug in multiple different model systems, neither of those worked very well. When you put them together, they can completely flatline the tumor, and that's what we want to prove in patients. So we'll be starting a trial in the next several months where we're going to be just increasing the dose. This is the dose escalation phase. We start at a low dose and we go up, daily dosing. Once we've hit a couple of doses where we're not worried about any toxicity issues, then we'll start a new arm where we'll go back to this dose, and then we'll add venetoclax based on its label. So it's a stepwise progression where we're going back and forth. And this is really what we're looking for, is can we show that in patients who normally were prescribed venetoclax, if they don't respond well, or if they come back after responding to venetoclax, will the combination of these two drugs prove synergistic? And that's the main objective of this trial once we get through the safety and toxicity issues. So that's an exciting program. There's a lot of competition in this field. Nobody has cracked the MCL1 yet. We're doing it from a gene regulation perspective. The final drug, and then I think I'll wrap up for questions, is called ME344. This is a really interesting problem. So cancer cells, as I say, they need a lot of energy. And typically, all of our cells in the body, they derive their energy from an organelle inside the cell called the mitochondria. It's the nuclear power plant of all of our cells. It kicks off what's called ATP very, very efficiently. It needs oxygen to do that. What you find as a tumor grows, the center of the tumor becomes what we call apoxic, or it doesn't get much oxygen because the blood vessels don't penetrate very well. So there's a whole new genetic program that kicks in that allows them to derive energy, not as efficiently, but to still derive energy and to maintain their cell growth, which is called glycolysis. Now, Avastin, was the number one selling biologic for cancer, blocks the ability for blood vessels to grow into tumors. Now, you'd think that would just starve the tumor, and that was the basis for the development of the drug. What in fact happens is it does starve the tumor for a little while, but then these hypoxic genes get turned off because the blood vasculature gets all normalized, and now they get oxygen again. And that's really why Avastin works, is because it normalizes the ability of the blood to get into these tumors, and now chemotherapy works really well. So Avastin has made chemotherapy better. But for us, what it does is it tips the scale back to away from glycolysis to force these tumors to use their mitochondria like they should, and our drug, which blocks the ability of the mitochondria to work, now synergizes, and these cells absolutely cannot escape. They're cornered and they die. And what we've just reported is that in a study in women with breast cancer, that we're just looking to see if they receive a dose of Avastin, 1 month of Avastin with a month of our drug, do we see biochemical signs that these cells are dying as we suspect? And the answer appears to be yes. So we're very excited by this. What's exciting also is it isn't just Avastin. There are multiple other drugs on the market, small molecule drugs that you can take that have the same effect as Avastin in multiple different cancers. So this could be a general way at going after what we call tumor metabolism. So I'll just go over that. This is the study that we're doing. We've just read out the first 20 patients on this trial, and we're now completing that trial. And hopefully we'll be reporting the interim data in this summer, and then hopefully the trial will be completed by the end of the year, and then we'll be announcing what we're going to do next. So in terms of near-term milestone, prasinostat, we'll be reporting the MDS data shortly, that we're going on to randomization if the data so suggest. ME401, we'll be reporting our ongoing trials in the summer with the initiation of the registration study. Versatility will be starting that trial shortly, and then ME344 will be reporting the data this summer. And I think that might be the last slide. So I'll stop and take any questions if there are any. Thanks for your attention.
This report brought to you by the Richmond Club, where investors and high-growth companies meet.